So, thanks a lot. It's a great pleasure to show some of the work we have been doing recently here. My name is Sebastian Munck. I'm standing here with let's say, a number of affiliations. So I'm associated with the VIB, the KU Leuven, but I'm also associated with Flanders BioImaging. So maybe to start there, what is Flanders BioImaging? We are a consortium of state-of-the-art imaging facilities. So that means we are connecting preclinical imaging, so that's, say, PET and CT and MRI scanners that are used in labs, not necessarily in the clinics, as well as high-end microscopy imaging, and we are located at all Flemish universities. And at most of these places we are also having both of these parts. Now, what does it mean? And maybe to give a bit more insight, from our perspective, we are running a facility. That means we have many researchers coming into our office demanding access to infrastructure, meaning microscopes. So we have high-end microscopes, and I try to avoid talking about big data, but it means we have machines that can spit out a lot of data. So while even maybe an individual image is still easily handleable in some tens of megs, but we can produce them on the millisecond level, and that means when we have only in our place some 12 of these machines and a handful of lower ones, this could easily amount to quite some massive amounts of data. Now, in addition, we obviously have the interest to automate these kind of things, so we run them over the weekend and all these kind of things, so that makes only things worse. So in a way, what we do, we are brokers of image data. So we try to obviously coordinate across our consortium and bring new technology developments. We are also part of a bigger infrastructure. There's an European infrastructure called VIB Bio Imaging, where we as Flanders BioImaging try to become an official node with more users, hopefully to come internationally to visit us. That's our new logo. But coming back to the data point, we are brokers for image data. So that means, to some extent, we are also part of a problem that we generate ourselves. So here, a lot of that, is obviously nice and good, and we want to mine that. We create a rich resource, but often, how do we handle that best? And we have, users, PIs that, have difficulties. PhD left 5 years ago. Where is that data actually really located? It somewhatly, creates a kind of a problem, and also kind of mindset where, oh shoot, do we have it correctly documented? What is the state of what we have, even if we find parts of it back? Data in that sense, let's say, has, as we say here, a dual nature. And, it, has, like pumping the oil in the image on the one hand side, we also create some kind of waste. Data, in that sense, evaporates, it's stored on USB hard drives that leave with the student, and this is a bit of a disaster, in short, right? So we as a consortium try to say, like, what can we do better? How can we do a lot of things? And, we would advocate, using containers, for example, that was mentioned before, for making your analysis more reproducible. But now coming to, how do we actually deal with the data? How do we store it? And how do we do this way better? Obviously, now staying in this analogy, we want to recycle, right? I mean, we want to get to a point where this is more usable. We want to use what is here called fair, so that data is findable, accessible, interoperable, and also reusable. And obviously, we need help for that. So here, the VSC comes in. Now, people have been publishing a lot on these. There is people that talk about the data lifecycle. How can we, support that from planning over collecting, analyzing, sharing, reusing, and so on. And there's also a lot of, or a few resources that would tell, how to annotate data good, when to associate which kind of information about how the sample has been created that is imaged, how the imaging itself has been moved or carried out, and so on. Now, in our environment here, together with with the VSC, we have an infrastructure at hand that we can use. And that's what enables this kind of workflow. And I will not talk here about all the individual icons that you see here. This is the environment that we have been using. Having been at a few of these research data management presentations myself from other colleagues, I often have the feeling that people talk about their favorite pet, a Pokemon you have never heard of, and where you say, like, what does it mean for me? And actually, do I need to Google this tool in order to know what it's about? So I want to go and talk with you about a journey that the data can take, and that we should here propose. As you see, there is the background of the different institutions where the instrument acquires a lot of these data. In the future, Globus has been named before. Obviously, we want to quickly and easily transfer the data towards Tier 1 data. Tier 1 data here, I will have a slide on this in a second, what this means. It's a data management tool that is loved by IT because it takes care about the data. It will provide you access to data that is mostly used very swiftly, while it will spin out some data that is old and not used onto some cheaper storage. So this makes a lot of sense. It's a bit like your photos on your phone, maybe. Those from 2016 are actually not really on your phone anymore. They have to be downloaded because you haven't accessed them for a while. Those from yesterday are still quickly available. It's a bit an analogy, but it's the same basic idea that's behind it. We obviously want to support that the people analyze these data. There's different opportunities. You just heard about the cloud, the high performance computing. It may be on your local desktop, never mind. But it needs to go back, the analysis, into the storage system. We need to keep track of that. And at the end, we want to publish these, right? I mean, we need to use the public repositories. We are here in the building of the FWO. It's very clear that they mandate us to actually put our data, once it's done and finished and we publish it, onto these institutional repositories or public repositories like the BioImage archive in our environment. Okay, so how does this scheme, and scheme is always nice, how does it translate to something that's a bit more real? So let's talk about it, and let's show it, and let's see if I can start playing that. So this is the first step. You see on the upper right corner where we are in the scheme that I just explained, right? So, and here we are just, uploading a few images through that Mango portal that has been named here onto iRODS. I know it's 2023 and I'm showing a movie of uploading data. That sounds a bit, simplistic, but it's actually very cool because we have here in this example just uploaded data onto a platform called Mango that allows us to manage the data on this iRODS background, where the data is managed in an IT system that me as a user don't want to really have much to do with, but it's very handy and actually allows us to have very cheap and competitive prices when it comes to storing our data on this. Now, there's other options as well, just to point it out. Globus has been named before. That's an general available software, and I'm just showing that because that's the information from the web page. It can be used in addition or as well for overnight take-ups of this data towards the iRODS data storage. Now, taking the next step, we want to associate metadata with the data that we have stored. And here you see my colleague Tatiana has created a very small and simplistic data scheme in this Mango platform, where we here describe that this has been acquired on what is called a spinning disk microscope and what kind of objective lens has been used. And what you see in the movie is that we now can look at search for these associated metadata. We have two types of metadata. Some is available already from the vendors. We are searching for this kind of information like pixel size and this kind of stuff. But we can also use that to find new metadata that we just added. This is obviously very important because at some later time point, like mentioned, when you need to find your data back, you want to know where it is, because in the iRODS environment you need to find your data through a query, through a search in the database, but it also means that you have to enrich your data such that you can handle it and work with it in the best way. Now comes the next step, because having the data, acquiring the data is only half of the deal, because obviously we want to analyze that. And here I'm showing an example where we collaborated with some people in Dresden that supplied us with some analysis tool, and we start on the VSC here a Jupyter notebook in order to analyze the data that you have just seen before that we have uploaded. So you see general Jupyter notebook. I'm not going into details, but as we scroll through it, you will see some of the data that we just uploaded loaded. Did this movie just get stuck? No, it's still running, I think. We just loaded it, and you can see there is some analysis tools that are used. There's some deep learning tool called CellPose and so on. You see the data that we just uploaded here in real life. We will analyze these, and while we go through it, we will generate some analysis plots and so on. And you can see that in a split second. There's some violin plots, how we extracted features from the data that is, so to say, on the tier 1 data stored. Okay. There's obviously different options to interact with the data. Open on demand has been just named before, and I'm just showing what we love to use in this environment, which is called the Bioimage Analysis Desktop. It's also called BAND, but it technically is here on the VSC a version from where Open on Demand is using our favorite imaging tools to open the image, as you have just seen before in the Jupyter notebook, how we can interactively work with those. Good. Now comes the interesting part. We have now analyzed the data, we have captured it. How does it actually get back into the storage system, into the tier 1 data? And for that, I'm trying to start here the movie again. We just injected a bit of code into the same Jupyter notebook that you have just seen before. So like this, we can interact back with Mango via a Python client, where we first check whether a file is actually there. We add metadata, we create a new folder where the analysis is stored. We have in that folder, we associate that with the analysis type that we have done. I've mentioned this CellPose deep learning algorithm for segmenting the data that is mentioned there, and we post the results in the type of CSV file. And in the movie you have just seen that we checked for that file and we searched for it, and we can associate it because we have the associated metadata there. So that's nice, because now we have closed the circle in a way, but to make it even more complete, we are now interacting with the radar, so the KU Leuven Research Data Depository, where we can actually fetch data from the Mango portal, and where we can actually see here a movie. There is a repository, there's first no data in there, so we actually locate them via Mango on the iRODS. We select a few data, and while we actually do this, we can here, obviously we don't need to publish everything that we want, but we can select for that. In the next second you will see that we actually publish that. We wait for an email to be sent by the radar back that the data is actually available, which is what you see here. And then we can check back, and you can see that the data is just uploaded together with our analysis if we want. And like this, we, so to say, have here closed the circle. That's just to show the radar as a repository of the KU Leuven. There's other ones obviously in Ghent and so on. In the future we want to use some also generic ones like the Bioimage Archive, which are hosted elsewhere. But overall what I try to convey to you is that we have here a data journey that we could show you, where we have a number of connected ideas or tiles that we can now use, where we have different possibilities to interact with the data, use Mango, use Globus to upload and search our data for the transfer, where we can associate metadata schemas and where we can, enrich our information of the data that we have uploaded, and where we can use different tools to analyze these, and then even close the step by depositing these files and analysis in our repositories. Taken together, this is here a proof of concept. It's open source in that sense. It's a complete workflow from acquisition up down to publishing. It's very flexible. There's an L missing, sorry for that. And it's content agnostic. So I'm showing this for images, but this could be anything. If you have your favorite collection of items from the Roman Empire, this would work likewise, right? So, and obviously in the future, we would love to see this kind of idea, picked up at other places as well, and obviously add more robust tiles to, enrich our environment. If you want to see more, there's actually a movie about Tier 1 data, where I'm featured here with my colleague from the VSC, Ingrid. There's also one how we run our imaging core. And I have a last slide to say on what is next, because we started this session with, how can ChatGPT help us? We have thought about how ChatGPT can also help us in this scenario that I just depicted. Because, we're talking about metadata, making life more reproducible. How can we, use that information? We used now to create data schemas on Mango, but there's also other sources. There is your lab notebook, there's the final manuscript that is all submitted, and we wanted to use the redundancy in these different sources, combine them, and leverage here ChatGPT to actually read that and correct for errors if there would be any. Because we know humans are terribly bad in copy editing. ChatGPT is a tool that can create structured output. So this is actually making a lot of sense. And here you see an example where we actually visualize the differences between the different sources. You can see that we actually failed here quite tremendously, because only the yellow ones, which are ones, would be actually good. But you can see that that's actually the state of where we are. And with that, I would love to thank you for your attention, and I guess we have a coffee break, no?